So the plan is to cover HIV over 4 lectures. The first 3, we're going to basically cover the majority of life cycle. Today, goal is to get through uncoding if possible. Monday, we'll talk about biosynthesis because reverse transcription is kind of a heavy load, so I want to make sure that we take that slowly. We'll talk about the mechanisms of expressing genes. Uses a couple of different forms that we've seen before. Talk about assembly, maturation, and exit, and actually probably by the end of lecture 3 we may even start to move into antivirals. And that's what I want to spend that lecture 4 on, is actually taking a look at specific antiretroviral drugs. Now, I haven't put together slides for them because the way I want to do it is actually just to kind of do review questions focusing on how these different retrovirals work. So consider this as kind of the build up to that. I was looking at a couple of papers yesterday, and the thing that I think is apparent to me, and some people I told this about, one of the reviews that I found is 30 years of HIV research, 624 references in this review. Okay? So if that tells you much of anything, I think what it signifies to me is we may not know everything about HIV, but we probably have more information about HIV than any other virus. We could probably take guesses as to why that might be. My personal opinion is probably because it was considered a life sentence originally, and we don't have a vaccine for it. Those are probably driving factors. The fact that it's a lifelong infection, and there are an incredible number of issues that roll into this that go beyond just the science. We could, just like with Ebola, we could talk about the politics, we could talk about social practices, cultural practices, we could talk about science denialism, which was really big in South Africa, at least through even the early 2000s, and has led to higher rates of HIV infection in South Africa, more so than any other country in the world. Okay? So there are a lot of things that are rolled up into this. We'll focus on primarily the science, of course, but if you have questions outside of that, feel free to ask, and I'll try to at least be somewhat informed about it. So here are the 4 objectives. Describe the life cycle, nothing new. Analyze, interpret experimental results to model and explain how HIV overcomes host hurdles. Again, a repeat of old ones. Evaluate how mutations and antivirals can affect the life cycle. And then Sabrina's favorite, hypothesize and develop a shit ton of experiments to test the effect of antivirals, etc. All right. So, obviously HIV, the cause of AIDS. Anybody know when this made an appearance in the United States, when AIDS was first known about, at least in the United States?
Like the 60s?
That's maybe a little too early. It's quite possible that there were cases then, but it just wasn't known.
70s?
Well, a little bit closer.
80s.
81 was really when we started noticing something. So this came from, there were a number of cases on the West Coast and the East Coast, San Francisco and New York City in particular, of a rare pneumonia caused by a fungal infection. And there were these number of cases that were popping up, and what they noticed in these individuals were they were all young men in their 20s or 30s. Probably, unfortunately, for the course of history, at the time, they were all homosexual as well. And so what that led to originally was the original name for AIDS was GRID, gay-related immunodeficiency. Okay? So it was, and with that came a lot of stigmas throughout the 80s. It took until about the 1990s when people started changing their mind. The term GRID was lost probably from the public about 85 or so. But AIDS predominantly in those early years was associated with homosexual males in particular. Nowadays, primarily associated more often with prostitutes and drug users, more than anything else. Drug users because of needle sharing, and then prostitutes because of unprotected sex. AIDS obviously is clinically defined, so it's defined by a drop in T cell number, and also an increase in HIV load. Somebody who is infected with HIV does not progress to AIDS right away. If an individual goes untreated and they are HIV positive, they'll probably progress to AIDS within about 5 to 10 years. And a very small percentage of the population, they will never progress. And most of the statistics that we're going to talk about here are about HIV-1. There is a second form known as HIV-2, which the progression of the disease is much slower. Sabrina.
What do you mean by HIV low?
Concentration, titer of the virus, yeah. So that'd be like 1,200, what would be equivalent to PFUs? That might actually be genome copies. I'm not exactly sure now that I think about it. These days, with the antiretrovirals that we have, if somebody can afford their antiretroviral drugs either through insurance or through their own doing, and the book gives some really good numbers on the cost of some of this medication, but if somebody can afford that medication, what we're finding is that individuals who are HIV positive are living as long, if not a few years longer, than the general public. The reason I think they might be living longer is because usually they adopt a healthier lifestyle in the long run. What it seems to be happening is that individuals who are getting the right care are dying from natural causes, or what we might consider to be natural causes, rather than AIDS nowadays. So here's just giving you a view of the worldwide distribution. North America, 2.3 million cases of HIV. This was as of 2013. Latin America, 1.6. North Africa, 230,000. Sub-Saharan Africa, 24 million. Okay? This is where the vast majority of the cases are coming from. And this is where, if we want to look at the social, cultural, political reasons, even economic reasons, this is an amazing case study. Okay, and there's a lot of good literature available on this as well. The U.S. has done some to try and help out with this. So for as much as you like or dislike Bush, one of the things that came out of his presidency was PEPFAR, which was basically it has created a pool of money from the U.S. to pay for antiretrovirals in sub-Saharan Africa. In the U.S., Affordable Care Act improved access to that medication within our country. Okay. There are hot pockets. Sorry, now I'm thinking of Jim Gaffigan from an old skit. But there are areas that we find higher prevalence than others, or we find actually these numbers are climbing at a much faster rate than others. Russia, rates are actually growing disproportionately compared to other areas. Southeast Asia, we see a similar trend as well. Okay. Numbers in sub-Saharan Africa and northern Africa are remaining, the rates remaining about the same, okay, as it was before. If we wanted to take a look at the natural history here, which we won't take a big look, but both HIV 1 and 2 originated in Africa and disseminated from there. We think, and I say we think because there is evidence to support that the earliest known case of HIV was in 1959. And this was some retrospective analysis. Somebody who I think had, I can't remember if it was malaria or some other infection, had gone in, had a blood draw. Well, they had, somehow there was remaining sample from this individual. Retrospectively, researchers went back in the 2000s and tested it, and it was HIV positive. So that was from an individual from a small village in Africa. Around 1959 was when that sample was collected. So that's the earliest known case. So based on predictions, what is thought is that HIV arose or made the species jump from chimpanzees to humans sometime in the 1950s, if not a little bit earlier. We also think that it's made more than one single jump. We think that there's been at least 2 events that have given rise to HIV-1 and HIV-2. One from chimpanzees, 2 comes from Pseudomangabes, which is another monkey species. Jonah.
Are those, like, monkeys the carriers of...
Well, they were carriers of what we call now simian immunodeficiency virus. So it's thought that, so they would have been the reservoir, and the thought is that these cases arose because, as we talked about with Ebola, chimpanzees and rodents and other monkeys would be bushmeat, right? And if the animal is darn near dead or dead on the floor of the jungle, it's easier to capture that than to capture a live one. So it's thought that individuals were infected through direct contact with blood, probably during the butchering process of the meat. Okay. All right. So that kind of gives a bit of a historical context of what we're talking about here. Here's kind of some of our basic facts. Single-stranded RNA virus. Of course, we know this to be a reverse transcriber, so we'll talk about that a little bit later. It's enveloped. Some literature refers to it as like icosahedral capsid. I don't really see it. It's definitely not helical. I kind of describe it as pleomorphic, and it seems to me to look more like a cone or candy corn, as I always describe it. It's got a number of different proteins. Inside the envelope, there are matrix proteins that are going to help to recruit the RNA polymerase, integrase other enzymes to the budding virus as it's exiting from the host cell. We'll talk about this a little bit later. There's 2 copies of genome. These copies are associated with reverse transcriptase, the enzyme integrase, the enzyme protease, and tRNA from actually us. This will serve as a primer for reverse transcription. Okay, so this tRNA, host-derived, actually becomes part of the viral particle and is essential to initiate viral replication. Why would this encode 2? Let me rephrase that. Why would HIV package 2 genomes per capsid? This is one of the few viruses that we see this taking place.
Could one be integrated and the other one be, like, just produced right away?
That's not a bad idea. That doesn't seem to be what happens, though. It's a good hypothesis, though. Take another stab.
If one is mutated, there's another one to...
Bingo. Exactly. This is one of those. This is a rare case here where we find a virus that technically, if you want to think of this in terms of genetic terms for a minute, it's diploid, right? If one copy of a gene is mutated, it's got a backup copy that can function. Okay, so and this probably in part, I can't tell you how this occurred evolutionarily, but you could argue that the evolutionary selection for this is because reverse transcriptase makes a lot of mistakes in the process of reverse transcribing. So as we go from RNA to cDNA during the process of reverse transcription, if RT makes a mistake on one of those 2 strands, well, hopefully the other one has that gene functionally encoded. So you've got basically heterozygotes in some ways. The tropism includes macrophages and T helper cells. The one thing that these have in common, more so the marker for T helpers, is what receptor? What do we find on every T helper? I don't know. That's what they recognize. It starts with CD.
4.
CD4. It was either 4 or 8, right? CD4 is the primary receptor. Now, things should start to make sense here in some way or shape or form, or at least we should start to be able to make connections. T cells are the primary host cell. We think, and just recently within the past year or so, researchers think they have found a subset of T helper cells that are the actual long-term reservoir for HIV. But we can start to make connections between that and the signs and symptoms of illness. Infection of CD4 positive T cells is going to likely ultimately kill them as the virus goes back into a lytic life cycle. That probably accounts for the reduction in CD4 positive T cells that we observe during this process. Okay. Obviously, if somebody is defective or unable to make CD4, then they will be immunodeficient. They've lost part of that adaptive immune response, to put this in as simple terms as possible. Let's see if there's anything else I wanted to... The other thing I want to point out real quick, and I'm going to draw this out just to give you guys a quick rundown of HIV. The graph that you will see in almost any textbook that talks about HIV is this x-axis refers to time, the y-axis refers to viral titer or concentration, and here's what you're going to find during a typical HIV infection. Somebody is infected within the first few weeks, we're talking like 6 weeks or so, they will develop symptoms. These symptoms are going to be flu-like, fatigue, weakness, maybe nausea, maybe. But obviously T cells are going to be numbers are going to go way down at this point. Macrophage numbers will go down as well. Then the virus goes dormant or goes latent for a long period of time. And this latency can depend on some factors. It can depend on the natural genetics of the host, can also depend on if the individual is being put onto antiretroviral drugs. It can also depend on the genetics of the virus itself. It's also okay, but over a period of time, and we're talking now years, we're going to see reactivation and slow increases in viral levels. Some point out here is where we find the development of AIDS. Okay. Go ahead, Grace.
So, if you don't catch this within like the first 6 weeks,
yeah,
is the chances that you're going to catch it later like less?
By catch it, do you mean identify it in the individual? I don't totally know. You would expect that somebody would have it might be harder to detect it down the road just because somebody wouldn't necessarily be keeping it in mind, right? Mm-hmm. What you would the things about HIV, this period of infection though, is it's a little bit longer than the flu, so you would hope that somebody would actually go in to see the doctor for this. Now, and there are a couple of ways that they now test for HIV infection. There are rapid tests, which actually they now sell for like at-home use even. You can do the test on your own if you wanted to, which are ELISA-based. They're detecting your antibodies to look for whether you have an immune response to HIV. The more sophisticated assays now are nucleotide ones, where they're looking for the genomic material inside T cells, for example. Jonah, did you have a question related to this?
I was just going to say, like, even after that, like, first, when it goes into that dormant phase, you can still test for the antibodies.
Yeah. Okay, thank you. And that's what I thought too. Mm-hmm. Now, if a doctor knows what they're looking for, they might ask also the right questions that would help to lead to the testing, right? So you would ask about sexual partners. You'd ask about drug use. You'd ask about the things that are consistent risks, right? And does your sexual partner have HIV? Somebody, you would hope, there are cases where this is not true, but that if you are in a monogamous relationship, you know your partner's status and they know their status as well, right? And that also could, so you would probably be informed enough to know that, like, oh my gosh, I'm developing flu-like symptoms, I should go get tested, right? There are some other things that we can talk about with, and we'll get back to this with treatment, but even there are not testing, but drugs to be used within the first 48 hours as well if you think you've had exposure. That can actually block the infection from ever occurring in the first place. We'll come back to this later on. Okay. All right, here's the genome, or sorry, here's the capsid. This is why I call it a cone or candy corn, because it is skinny on one end and a bit fatter on the other, like a candy corn. Obviously enveloped. The envelope proteins, we'll talk about specific proteins as we go along, but the GP120 and GP41 create the glycoproteins, so these are our spikes. GP120 is the true spike. GP41 actually helps out with fusion. We have the matrix proteins. I'm not going to ask you about the specific names, to be honest, but there are a number of matrix proteins that contribute to making up this inside here, helping to create a scaffold to pull everything together. We've got capsid proteins like P24. We've got 2 copies of genome, and with that genome are associated reverse transcriptase, integrase, tRNA lysine, protease, etc. And I'll be honest, I don't know what the CYP-A protein or ICAM are doing. I'll look and see if that's something we need to consider. Okay, so that's the basic structure. Question? No? Okay. Here is the genomic structure. In all retroviruses, HIV, Rous sarcoma virus, HTLV1 and 2, they all have a common set of genetic features. They all have what we call these, long terminal regions, LTRs. They all have GAG, POL, and ONV. GAG encodes the matrix and the nucleoproteins. So you can think of GAG as an encoding structure of the viral particle. POL is short for polymerase. It encodes the polymerase, also encodes other things that are going to be required for getting the viral genome into the host. So it encodes for integrase, as well as the protease, which we'll come back to towards the end of replication. And then ONV encodes for the envelope proteins, GP120 and GP41. We're not going to talk just yet about the transcription of these, but I just want you to know that these are there. There are a number of others, what are termed as accessory proteins for HIV that have a number of different roles. And these include proteins like VIF, VPR, TAT, NEF. Okay, we'll see a couple of these. We will take a look at. It seems like we're missing one here, and I'm not sure where it is. But no, the VPN wasn't what I was looking for. But there's one known as Rev, which we'll come back to later on as well. But we'll look at Rev and TAT. These are both going to be involved in biosynthesis. And then these other ones like VIF, VPR, VPN, NEV, those are all involved in modulating the cellular response. Okay, so not only the innate response inside of cells, but also actually affecting the adaptive immune response. So let's take a look at that real quick. Sabrina.
What did you say LTR was?
Long terminal repeat. And we'll get to how this is formed later on, but the 5' end of this LTR actually serves as the promoter for transcription. Okay. So TAT and REV, as I mentioned, these are going to be involved in regulating biosynthesis. We'll come back to their specific roles later, but in brief, REV helps to bring unspliced RNA out of the nucleus. So this is going to be used actually specifically to take full-length HIV genomes and make sure that they're exported from the genome without being spliced. TAT, on the other hand, is going to activate transcription. It's going to help to recruit RNA polymerase and other transcription factors to activate the process of transcription. Okay.
Can you say Rev one more time?
So REV's role, it's going to bind to the viral mRNA. And if you think about it real quick, I guess I need to describe one piece of this. HIV will perform alternative splicing as one of its mechanisms to make mRNAs. Well, that's great, but you also need full-length genome that's unspliced. REV helps to make sure that that full-length unspliced mRNA makes it out of the nucleus and into the cytoplasm. Because apparently nuclei don't like unspliced mRNA exiting the nucleus. I don't know why. I have to figure that out.
So just make sure that the mRNA makes it into the cytoplasm?
Yep, exactly. Helps with that nuclear export. Sabrina.
So what would that unspliced mRNA go for, like packaging new variants or
Exactly. So it would get packaged into new variants that are forming.
the genome?
Yeah, exactly. I'm not really concerned with you knowing the function of, or memorizing the function of NEF and VPR and VPU and VIF, but I do want to kind of point out a couple of things here about them. Note that 2 of them are involved in downregulating CD4. What's the benefit of this? CD4 is the target. Why repress the expression of the target? And you could think of maybe a couple of potential answers for this.
CD4 would try to destroy infected cells.
So one would be the immune response argument, right? That by repressing CD4, that prevents those T cells from activating or helping out with the adaptive immune response. That's absolutely one potential argument. You could argue that there's a second reason here. What happens when you repress the receptor for a virus? What does that do to the life cycle?
Prolongs
It could prolong it. Let's ask this a slightly different way.
By repressing the receptor?
Oh yeah, because CD4 is the receptor. So let me ask this a slightly different way. You have a T cell. It has CD4 on its surface. You have another T cell. It does not have CD4 on its surface. Which of these 2 can be infected by HIV? The one with CD4, right? This one cannot be infected. Why would blocking infection be advantageous?
For the host or the virus?
For the virus.
Isn't it already inside when it
Good. It's already inside. It's already there. Virus is already inside of these cells, and it's repressing CD4 expression once it's there. So why do that? Exactly. It actually contributes to the spread of infection into new host cells. It's basically like a marker saying, hey, I got this under control. We're already good. All right. Go ahead, Jonah.
So what's the gene that represses the CD4 expression?
So there's a couple of them. There is NEF, and the other one is VPU. Now, there are a couple of other ones in here, like VPR and VIF are going to be required for replication or infection. VIF, for example, blocks a protein known as, and I'm just telling you this to give you context, I'm not asking you to memorize this, blocks a protein known as ApoBec3G, which has actually been intensely studied with respect to HIV infection. ApoBec3G actually resides in the cytoplasm. It removes amine groups from cytosine. And in doing so, it converts the cytosine to a uracil. So instead of having a CG base pair, you get a GU base pair, which when you get replication would lead to UA or GC. So it actually is used to mutate the viral genome. Well, VIF blocks that process and ensures that the viral genome isn't going to be hypermutated. Okay, so that's been a big area of study. Trying to use our own innate immunity as a way to block infection. All right, your turn. Here's the life cycle. Let's do the same thing we did with Ebola. I want you to take this, and I want you to take about 5 minutes or so. Go through the steps, describe what's its mode of attachment, what's its mode of entry, what's its biosynthesis, so on and so forth. All right? Go. Factors for reverse transcription like that tRNA lysine, etc. We've got some uncoding. The genome's going to make its way through a nuclear pore, and then we have integration. Go ahead, Sabrina.
So by the time it goes through the nuclear pore, that's cDNA?
It is.
Okay.
And actually, by that point, it's already double-stranded cDNA. Yeah. Go ahead, Jonah.
Is there a reason why it's shown as a circular piece of DNA in that second?
This here,
Yeah.
I don't think it's meant to be circular. I don't know. I just noticed that myself. I can't tell with the resolution if that's meant to be circular or.
It's got some like little purple things.
Yeah, and I thought at first that might be like the reverse transcriptase, but I don't know. It is not. Oh, you know what that might be? I don't think it's representing that it's circular. What I think it's representing is the integrase will bind to the ends, and the integrase kind of interacts with itself, so it might represent that it's not really circular, it's just a linear piece that's kind of looped back on itself with the integrase.
Gotcha.
So what the integrase is going to do is help with this integration process and help to splice that DNA randomly. We think 15 years ago it was random, then 10 years ago they found hotspots where there were more frequent insertions. I don't know if they know what contributes to that, but the integrase is going to help to ligate everything back together and put two copies in different locations in the host genome. You can consider the reverse transcription here as part of biosynthesis. Okay, if you're trying to mentally group things together, consider it as part of biosynthesis. But then once in here, this is where we will get out what we have termed biosynthesis throughout the semester. Replication here is driven by, depending on how we talk about replication, right? I guess technically it all is driven by the host, but if we wanted to know which protein, well, if it's during cell division, if it's during the latency phase of HIV, it's just replication, so it's driven by the host DNA polymerase. If we're talking about producing virus, so lytic replication, that requires RNA synthesis, so it would be the RNA polymerase. What this process here is trying to show is that oh, go ahead, Jonah.
Do T cells divide, or are they just made by like red bone marrow?
So they will divide at some low rate, but they'll proliferate when stimulated. That's where they really do divide.
Like during infection?
During an infection, for example. What this process here is trying to show is that this virus is actually going to exit through exocytosis, but it's going to be through the structural proteins are going to be made in the ER, they'll get modified in the Golgi, and what this is showing is that they will prevent or be present on the surface. So these get made in our exocytost, get embedded on the surface here. We get matrix proteins, we get capsid proteins, all of that assembling, and what is this actual exit process here? That's budding. Okay. One of the things that's really odd about HIV is it doesn't actually undergo maturation until it's exited completely. So there's a reversal of order here, or a change in order. Instead of the last few steps being biosynthesis, assembly, maturation, and exit, it goes biosynthesis, assembly, exit, final maturation. It is thought that the reason why that occurs is so that HIV doesn't reinfect the same cell. So it actually moves away from that cell before it is active and ready to infect again. Sabrina.
So when it integrates into the host genome and then it undergoes biosynthesis, are the host genes also being replicated and transcribed, or is it just the...
No, it would be, if it's part of the genome, it's going to be anytime the host undergoes S phase, the viral genome is replicated along with the host genome. So this process here, during the replication, during latent replication, it'll be dictated by the host. When you're talking about transcription of the full-length mRNAs or those spliced ones, this would occur as the host is transcribing its own genes as well. Does that answer the question? Okay. Jonah.
So both copies of the genome are integrated. In this picture only one is
It's showing it for simplicity's sake, yeah.
Okay. Can they both be active in...
Yes.
Okay.
And hopefully you want both of them to be active, right? Just from the perspective of, again, you're hoping that one or the other is going to produce the functional proteins that allow for that exit strategy. Go ahead, Grace. I'm just touching this so it doesn't turn off.
Do you know why it goes through the ER and the Golgi?
I think that's because and this is something we haven't brought up, but any protein that is membrane bound actually is going to be made on the ER because it gets embedded in the membrane in the ER. Then it undergoes modification like adding glucose or other sugar moieties onto it in the Golgi, and then it's exocytosed to be presented on the surface.
Okay.
So we would actually see this process occurring with any envelope virus. It's not as if those transmembrane proteins are being made in the cytoplasm and just find their way to the cell surface. They've got to go through this process.
So, just to clarify, it's like those proteins go out of the cell through exocytosis
Yeah.
and then meet up with the other proteins that are budding?
Yep, so it would meet up with these cytosolic proteins,
Okay.
and then that would force the budding to occur.
Okay.
Sabrina.
So does the exocytosis of the glycoproteins drive the self-assembly?
It can help to drive the self-assembly, yes. Yes, it's actually kind of a precursor because the internal domains of these, or the regions that are hanging on in the cytoplasm, those are going to serve as anchors for the matrix protein to bind to and accumulate on. Jonah.
Would you find those spike proteins like all over the surface of T cells?
Absolutely. Now I say that, but there might, theoretically, yes. If you look in the literature, there are sometimes you will find localization of these, and it's not really defined well what that localization is. I'm thinking of an example for EBV, Epstein-Barr virus. It's enveloped. Its glycoproteins locate to what we call lipid rafts. I'm not going to go into detail of what it is. It's something that's a little bit different than what we consider to be the regular phospholipid. But we might find localization to particular regions of a cell. But for the purposes of what you guys have learned in terms of cell biology, yes, it's all over the place. Sabrina.
When it says released by lysis at the bottom, what does that mean?
Well, I'm guessing that means that the cell is actually rupturing to release the virus, but that doesn't really look right to me because it's a budding process that they're releasing. I'll be honest, I don't know.
Cause I'd have.
You could email the author of our textbook who's in Oshkosh if you want to go visit her over the weekend.
Oh.
No? Okay. I hear she's kind of mean anyway.
Oh.
No, go ahead. This is another
Okay.
picture of the same thing.
So it looks like there's like a co-receptor in their attachment thing, CCR5.
Ah, CCR5. Can we transition to that?
I guess.
I think that would be a great idea. That is the co-receptor. Okay? So HIV uses two different receptors. The initial attachment is to CD4. And that is exactly what GP120 is going to do. So the interactions here, again, protein interactions drive change in shape. All right. I'm going to go through this quickly because we're running out of time and we'll pick up here on Monday. But in brief, GP120 binds to CD4, and note what it's saying here is that these variable loops that are on GP120 go from kind of a closed conformation to open up once they interact with CD4. That exposes a second binding site. That second binding site is what binds to CCR5 or CXCR4, depending on the cell type you're talking about. So macrophages have CCR5, CXCR4 is found on T cells. When that second binding occurs, an additional change in shape happens. This allows for GP41 to go from a closed conformation to open up, basically grab a hold or fuse with the plasma or host membrane, and now pull them back together. And that's how the fusion event occurs. So it's a three-step process. Bind CD4, bind the co-receptor, GP41 involved in fusion. Okay. We'll leave with that. But Jonah brought up asked me a question about if I've heard about certain individuals that have mutations in CCR5, and yes, this is a really hot area of HIV research, or at least was even 10, 15 years ago. So, over the weekend, take a look at, just type in CCR5 HIV, and you'll come up with a bunch of literature or a bunch of information. Another one you may want to look up, Berlin patient. There's an individual known as the Berlin patient, and another name, Timothy Brown. Okay? Look those up, and that will give you some information on the importance of CCR5 here. All right. Have a great weekend. Don't forget to submit your posters if you haven't already. And send me a copy too by email. That's perfectly fine. So I can get one from everyone.
Is that?
Yeah.
We're gonna go pick up our